Hi everyone, this is a lecture on Chapter 7, Molecular and Cellular Radiation Biology. We're going to talk about the branch of biology that concerns itself with the effects of ionizing radiation on tissue, on living systems. This is going to include the sequence of events occurring after the tissue has absorbed ionizing radiation energies, the action of the tissue or the living system to make up for those consequences of absorbing the energies, and the injury to the living system that may be produced. Okay, ionizing radiation damages the system by ionizing atoms, and ionized atoms will not bond properly in molecules. Okay, so when irradiating tissue, the response to the radiation is determined by the amount of energy deposited per unit mass, so the doses in grays. And during experience, to test the response of tissue to equal doses of radiation to equal tissue specimens, the results vary depending upon the physical factors. Okay, physical factors are charge, mass, amount of energy that's associated with different types of radiation. And these attributes determine the extent to which the radiation can damage or transfer energy into the biological tissue. Okay, these characteristics can affect the amount of biological damage. And the radiation characteristic that is most important in determining cell damage is the rate at which the radiation deposits its energy. And this is called LET, linear energy transfer. It describes how the ionizing radiation energy is deposited along a tract or path of tissue. Okay, LET has units of kilielectron volts per micrometer. As the radiation passes through tissue, it deposits energy and produces ionization of the cells in the tissue. Different types of ionizing radiation have different LET values. So there's high and there's low. Okay? High LET radiation passes through tissue. It deposits large amounts of energy in a short distance. And likewise, high LET radiation has a greater biologic effect. So if it's depositing large amounts of energy in a short distance of tissue, it's going to have a greater damaging effect. It has very little penetrating ability because it loses all of its energy in a short distance. Okay. Now the low LET radiation is very penetrating because it's spreading its energy over large distances. So there's little chance that the low LET radiation will deposit more than one ionization in any one cell because it's got high energy, it's penetrating quickly, it's moving over a longer length of track. Okay, here is a slide of an electron with 350 kilielectron volts. So remember, the high LET radiation has real values that they've measured from about 10 to 200 kilielectron volts per micron. So this is a slide that overemphasizes what high LET can do. It is bouncing all over the place. As soon as it enters the tissue, it is interacting numerous times, boomeranging back and forth. And at each interaction and ionization that it's making, it's losing its energy. Okay, so that's just an example. Okay, this next slide just shows you the types of low, which are gamma ray and X-rays. And remember, those have a value from about 0.2 to 3 kilielectron volts per micron. Okay, and then the high LET radiation, which has that value once again of 10 to 200 kilielectron volts. And the types of that is alpha particles, ions of heavy metals, things like that. Okay, our next concept is relative biologic effectiveness, or RBE. It describes the relative capabilities of radiation with differing LETs to produce a particular biologic reaction. Biologic damage produced by radiation escalates as the LET increases. And identical doses of radiation of different LETs do not render identical biologic effects. So they had to figure out a way to be able to say this amount of radiation of this type produces this effect. And since we have different types of ionizing radiation, they needed some way to determine how much is a bad thing of, for instance, a high LET like alpha particles compared to a low LET like X-ray. They did this by making a test, and the reference radiation that they used is 250 kVp, and the reaction is what is produced by a dose of the test radiation delivered under the same conditions. And looking at the next slide, looks like this one, shows how they can use that number to have a quantifiable effect that they can use to explain different types of amounts to produce the same reaction. So, for example, a biological reaction is produced at 2 grays in tissue of a test radiation, and it takes 10 grays of 250 kVp X-rays to produce the same reaction. So what is the RBE of the test radiation? So, and this is the equation, 10 over 2 equals 5. So the conclusion is the RBE is 5, which means that this test radiation or whatever ionizing radiation that they were trying to figure out that they were using is 5 times as effective in producing the biological reaction as the original 250 kVp X-ray. So they also figured out that this is not practical. This number is not practical for specifying radiation protection dose levels in humans. So to overcome this, they came up with the radiation weighting factor, so the WR. And that's what we used last week in our equations, and they're the same values. Okay, so we're going to be using the WR. Just know that the RBE was their way of coming up with that radiation weighting factor. Okay, so X-ray and gamma rays, the WR is 1. And alpha particles, it is 20. Okay, so remember those numbers. The next concept is the oxygen enhancement ratio, OER. Okay, the OER is the ratio of the radiation dose required to cause a particular response in a tissue. One is in an oxygen-deprived environment. The other one was in a regular, normal, oxygenated environment. And they came up with this ratio. And what the bottom line is they came up with was the oxygen fixation hypothesis. It determined that there is certain types of ionizing radiation where it doesn't matter if there's oxygen in the tissue or not. There's lasting effects, period. The damage is done. Compared to low LET radiation that can go through a tissue that doesn't have a lot of oxygen, and that repair can be fixed by the cell itself. It can be repaired. And versus low LET radiation that can go through a tissue that has oxygen, very oxygenated tissue, and when any damage occurs in that, then it's lasting. It can't be repaired. So this phenomena has been called the oxygen fixation hypothesis. Okay, so those, just review that. That's pretty much all we need to worry about on that. Okay, this is the oxygen enhancement ratio. This is the radiation dose required to cause a response without O2 over the dose required to cause the same response with O2. Okay, called the oxygen enhancement ratio. Okay, next we're going to be talking about the molecular effects of irradiation. This is where we get into the molecules inside the cells, and when one of those molecules is damaged, what effect it's going to have on our cell. And as a result, anything done at the cellular level shows up in our organic system. So any visible radiation injuries that you can see or that we can quantify with CT, MRI scans, whatever, of living systems at the cellular organic level, they always began with damage at the molecule, at the molecular level. So molecular damage results in the formation of structurally changed molecules that impair cellular functioning. That's when things start to break down. Okay? Okay, so the effects on somatic and genetic cells are germ cells. Because energy from radiation can alter the cell's molecules, That exposure can disturb the chemical balance. Ultimately, the way it operates, it can lead to cell death, which can lead to tissue or organ damage, so the body's processes are disrupted. If radiation damages the germ cells or the genetic cells, the damage is passed on to future generations. So there are two types of ionizing radiation interactions on a cell, either direct or indirect. So it depends on where the interaction is taking place in the cell. So if it interacts with a macromolecule, a vital molecule, such as RNA, DNA, proteins, enzymes, this is called a direct action. If it's indirect, it is interacting with water. And when it interacts with water, it is always a multi-stage process that involves free radicals. Okay, so because the human body is 80% water and less than 1% DNA, essentially all the effects of low LET radiation, X-ray and gamma rays, in a living cell result from this indirect action. Okay, we're going to stop here and continue with part 2 on getting into how the direct and the indirect actions break down and what they look like inside the cell and what damage they can do. Okay?